Hello dear students, welcome back to my channel. I am Professor Namrata Burache. Today we are going to continue the next topic of 11th standard, that's a cell structure and organization. In this, first of all we see that's a what is meant by the cell. The cell is the structural and the functional unit of all living organism. The cell, it is a first time discovered by Robert Hooke. Anton von Leeuwenhoek first observed and described a live cell. The invention of compound and electron microscope revealed the structural detail of the cell. The Sheldon proposed that the plants are composed of a cell. So, the Sheldon and the Schwann proposed the cell theory. What's a cell theory? The Schwann in 1839 reported that the cell had a thin outer layer. That thin outer layer, it says as the plasma membrane. He also found that plant cells have the cell wall. He proposed the hypothesis that the plant and animals are composed of cell and products of cell. Afterward, Virchow in 1855 explained that the cell divided and the new cell are formed from the pre-existing cell. So, he modified the cell theory. So, what's the statement of the cell theory? The first statement is that is all the living organism it composed of cell and the products of cell. Second one, all the cells arise from the pre-existing cell. Next one is total activities of the cell are responsible for activities of organism. Cell shows the transformation of energy. Cell contains the nucleic acid, that's the DNA and the RNA in the nucleus and the cytoplasm. Afterward that the cell contains the basic component. Means each and every cell contains the basic component. In that the first one is cytoplasm and the second one is the ribosomes. The cytoplasm, it is the semi-fluid matrix where cellular activities and the chemical reactions occurs. This keeps the cell in the living stage. This is a very important point in case of the cytoplasm. Second thing is that is there is the presence of ribosomes. These ribosomes, it is a non-membrane bound cell organism found in cytoplasm, chloroplast, mitochondria and on rough endoplasmic reticulum. Means on the rough endoplasmic reticulum on outer side there is the presence of the ribosomes. So these ribosomes, it is a non-membrane bound organism. Means it is found in all these organisms. It is present in the cytoplasm, it is present in chloroplast, mitochondria and on the rough surface of the rough endoplasmic reticulum. As the cell shows the various in size and the shape. Means the cell size varies greatly in the various plants and animals. The smallest cell size can be seen in case of the mycoplasm. Its size, it's near about the 0.3 m in length. The bacterial size cell, it's near about the 3 to the 5 micrometers in length, while the largest size cell which is seen in case of the egg of ostrich. Okay, afterward that the longest cell, it says as the nerve cell. The size of bacteria, we already know that it's a 3 to the 5 m. In case of human, RBC cell, that is a red blood cell, it's near about the 7 m in diameter. In addition to this, the cell, it does not having the typical shape. Means there is no typical shape of a cell. The cell, it may be disc-like, it may be polygonal, it may be columnar, it may be cuboidal, may be thread-like, or it may be the irregular in shape. The next phenomena we see, that's the structure of the prokaryotic cell. What's the characteristic of the prokaryotic cell. In prokaryotic cell, they have no membrane bound nucleus and the organelles. Means there is absence of mitochondria, chloroplast, endoplasmic reticulum, and the well-developed nucleus is absent. They include This prokaryotic cell includes the bacteria, blue green algae, mycoplasm, then the pleuropneumonia like the organism, that is a PPLO. They are generally smaller and multiply more rapidly than the eukaryotic cell. They vary in size and the shape. So this is the bacteria. Bacteria, it is present in the various forms. It is a green bacteria, that is says as the blue green algae, the cyanobacteria. Then there is a presence of mycoplasm. So this bacteria, it's a various forms of the bacteria which is included in the prokaryotic cell. Next one, the bacteria, it's having the 4 basic shapes. The first shape is the bacillus. Second one is the coccus, that is the spherical one. Vibrio, it's a comma shape, and the spiralium means the spiral. So this one is the spiralium. This one is the comma shape. This one is the bacillus, and this one is the spherical shape. So the shape of the bacteria is also varies. The next one about the organelles which is present in the prokaryotic cell. The first organelles which is present, that is says as the cell envelope. First one is the cell envelope. There is a presence of the 3 forms of the cell envelope. Outer one, it says as the glycocalyx. Second one, it says as the cell wall. And the third one, it says as the plasma membrane, that is innermost is the plasma membrane. So it says as the cell envelope. Second one, there is a presence of the mesosomes. Next one is there is a presence of nucleoid. Then the next one is there is a presence of the flagella, then the pili and the fimbriae. The next one is ribosomes is there and the inclusion bodies. So we see the one by one. The first one is the cell envelope. The cell envelope, it is the chemically complex protective covering. It consists of the 3 layer. Outer layer, it says as the glycocalyx. Second, it says as the cell wall. And the third, it says as the plasma membrane, that is the innermost layer. The first one we see, that is the glycocalyx. So what's a glycocalyx? This glycocalyx, it is the outermost layer. This layer differ in thickness and the chemical composition in the different bacteria. It is mainly of the 2 types. First one is the capsule, that is this capsule one, and the second one, it is the slime layer. This capsule, it is comparatively thicker, tough, and more compact than the slime layer. The second one, it's a slime layer. This slime layer, it is a loose sheath which is more diffused and the less compact. Both these layers, it is mainly made up of the polysaccharide, but may contain the protein also. The second component we see, that's a cell wall. The cell wall, it is the second component. This one, okay. The cell wall, it is the second layer made up of the peptidoglycan or the murin in eubacteria, and in case of the arachnoid bacteria, it is consist of the pseudopeptidoglycan. This pseudopeptidoglycan means arachnoid bacteria, it is a gram-positive bacteria, and the peptidoglycan and the murin, in case of eubacteria, it is a gram-negative bacteria. This cell wall, it is function as it gives the shape to the cell and provide a structural support to prevent the bacterium from the bursting or the collapsing. The third innermost layer, that is says as a plasma membrane. This plasma membrane, it is a innermost layer. It's a innermost layer. It's a innermost covering made up of the lipid and the protein. This is a very important part. It is made up of the lipid and protein. It is a site for respiration, photosynthesis, synthesis of lipid, synthesis of cell wall and the component. This plasma membrane, it's a semi-permeable in nature and interact with the outside. They are structurally similar to that of the eukaryote. The cell envelope, it's a very important part in case of the cell envelope. Based on the type of the cell envelope and response to the Gram staining, the bacteria, it's of the two type of bacteria. The first one, these two type of bacteria, it is the first time discovered by the Christian Gram. It's the first time discovered by the Christian Gram in 1884. These two type of bacteria, the first one, it's a Gram positive bacteria, and the second one, it's a Gram negative bacteria. What's a Gram positive bacteria? That is, this bacteria stained by the crystal violet and washed with alcohol, so it retained the stain. It's retained. The second one is a Gram negative bacteria. This bacteria stained by crystal violet and washed with alcohol does not retain the stain, and it gives the mechanical strength to the cell. Afterward, the next phenomena we see, that is in the cytoplasm, in the matrix, that is a semi-solid, semi-fluid crowned substance means the matrix, which is present inner to the plasma membrane, inner side to the cell membrane. It is consists of the varieties of organic and the inorganic component. It does not show the streaming movement. It does not contain the membrane-bound cell organisms such as the mitochondria, endoplasmic reticulum, Golgi bodies, lysosomes. But there is the presence of the mesosomes. So what's the characteristic features of these mesosomes? The mesosomes, it is the formed by the infolding of the plasma membrane, that is the cell membrane. It is present in the three forms. First one is a vesicle. So this vesicle, it look like this. It's an oval globular structure. It may be the tubule, so it's a tube-like a structure. Okay. And the third one, it's a lamellae. So this lamellae, it look like this. Okay. Look like as a lamellae. Means the mesosomes, it is present in the three form. This mesosomes, it is function as the cell wall formation. Second, for DNA, this chromosome replication. Then for the distribution of chromosome to daughter cell, for respiration and the secretion, to increase the surface area of plasma membrane and the enzymatic content. These are most important functions which is worked out by the mesosomes. The next organism which is present in it, that is says as the chromatophores. These chromatophores, they are present in the photosynthetic bacteria and the cyanobacteria. They contain different types of the pigment like the bacteriochlorophyll, bacteriophyophytin, and the carotenoid. So these chromatophores, it is present in the bacteria, that is the pigments. It's present in the various forms of the pigments like the bacteriochlorophyll, bacteriophyophytin, that is a brown color, bacteriochlorophyll, it's a green color, and the carotenoid, the orange color. So this chromatophores is a very important role in case of the bacteria. The next phenomena we see, that's the nucleoid. This nucleoid, it is form of the non-membranous, piece-necked, circular genomic DNA, single chromosome and the genetic material and the protein. Many bacteria have the small circular DNA. This small circular DNA, it called as the plasmid. This plasmid, it is a circular double-stranded DNA molecule having the ability of the self-replication. This plasmid, it's outside the genomic DNA. Means this one is a genomic DNA, and outside to the genomic DNA, there is a presence of the circular double-stranded DNA, that is says as the plasmid. It gives some unique phenotypic characteristic, that is resistance to the antibiotics to the bacteria. The next one, it's a flagellum. The flagella. These are the thin skin. Filamentous extension from the cell wall of the motile bacteria. Their number and arrangement are varies in the different bacteria. The bacterial flagella, it consists of the three parts. First one is the second, and this one is the third part. Okay, that is, the first one is a filament, second one is the hook, and the third one is the basal body. The filament, this filament, it is the longest. One is a filament. The filament is the longest portion and extends from the cell surface to the outside. Here these are surface structure that have no role in the motility. The pili are the elongated. So this is that's the okay. It is a elongated. They are the elongated tubular structure made up of the special protein, that is the fibrini are the small bristles like a structure. Okay, these are the small bristles like a structure. Bristles like fibers sprouting outside the cell. In some of bacteria, they help to attach the bacteria to rock in stream and to host. Next important, very important part is the ribosomes. These ribosomes, they are associated with the plasma membrane of the prokaryotes. They are about 50 nm by 20 nm in size. They are made up of the two subunits. The first one, it's a smaller subunit, and the second one is a larger subunit. The smaller subunit is a 30S and the larger subunit is the 50S. They together forms a ribosomes. It's a 70S ribosomes. What's a S? S is the sedimentation coefficient. That is a measure of density and the size. The first time discovered by the Sedgwick. So what's the function played by these ribosomes? Ribosomes, it is a site of protein synthesis, means the translation phenomena which is carried out by the ribosomes. Several ribosomes may attach to a single mRNA. So this one is the single mRNA, and the several numbers of ribosomes, it is attached on a single mRNA to complete the process of the protein synthesis. When the several ribosomes, it is attached on the single mRNA, then it is said as the polyribosomes condition. The ribosomes of the polysomes translate the mRNA into the protein. Next one is the inclusion body. This inclusion bodies is a very important part. These are the free storage granules present in the cytoplasm. These are the non-membranous, store the reserve food material seen freely in the cytoplasm of the prokaryotic cell. There is a two forms of the inclusion body. It's a organic and the inorganic. Organic inclusion body, it's a cyanophysin starch granules, glycogen granules is there. And in case of inorganic inclusion, there is a presence of phosphate and the sulfur granules. There is a presence of the gas vacuoles. What's a gas vacuoles? They are found in the blue, green and the purple and the green photosynthetic bacteria. Hope so you understood it. Thanks for watching my video. Stay tuned to my channel. Thank you.